Hi, my name is Nadine Aranis. I'm a physical therapist at Clinical Eman in Santiago, Chile, and I'm currently at TA1 at PPCR. So I'm going to present you today a rapid five about basic study designs, and I have no conflict of interest related to this presentation. So study designs are a set of methods and procedures that are used to collect and analyze data in a study. And broadly speaking, we can have many ways to divide epidemiological studies, but one simple way is in observational versus interventional studies. Observational studies, which you're going to check on later during the year, are non-experimental ones and can be descriptive or analytical, depending in if they have a comparison group. While interventional studies are going to be experimental ones that can be randomized or non-randomized. If they are not randomized, they are also called quasi-experimental studies. The most important thing is that study design will help you delineate the methodology of how to obtain the answer to your research question. So even though we know that the strongest level of evidence is from randomized controlled trials, we might not have to use that approach for every single clinical research question. The main characteristic of the experimental designs is that it allows the investigator to manipulate the intervention or treatment variable, also called independent variable. And as the assignment is random, you can also control for co-findings, so getting a higher internal validity. Observational studies do not allow the investigator to manipulate the intervention, and in this case, it's actually called exposure. So the relationship between the exposure and the outcome, which can be the disease, is usually confounded by other variables. Getting into the experimental studies, we can have parallel and non-parallel group designs, depending if the subject is assigned to one study arm or not. The first ones that we're going to check on is the parallel design group, which is the most commonly used. And I will show you here some examples so that you can understand it better. If we have one independent variable, this means basically one treatment in different formats. We can compare intervention versus placebo, for example, administering exercise therapy versus placebo intervention in patients with chronic pain due to knee osteoarthritis. The second option could be comparing intervention versus an active agent, which is usually the standard of care, for example, exercise therapy versus a non-steroidal anti-inflammatory drug for the same population, or making a multi-group design, which is going to be comparing an intervention versus a control 1 or a control 2, or could be a placebo. If we have two or more independent variables, this means we have two treatment in different formats. We will probably do a factorial design. And the most common one is a 2x2 factorial design. For example, comparing manual therapy versus exercise therapy, then you compare both of them and compare it to usual care for patients with knee osteoarthritis. So sounds great because you can simultaneously test two or more hypotheses, but you have poor power in this kind of designs, especially to detect an interaction, because if that interaction is actually present, the factorial design can produce misleading results. To finalize this rapid five, we have the non-parallel design group that are basically when each subject is assigned to one, they are not assigned to one study arm, rather they are assigned to repeated measures. In this case, if they have one independent variable, we have the crossover design. And I leave you an article here as an example. That could be, for example, testing a test group first that received Tuina, which is a Chinese massage treatment for 4 weeks, and then received health education intervention for another 4 weeks. And in parallel, you will have a control group that received health education intervention for 4 weeks, and then received Tuina treatment. So they kind of changed the treatment and the intervention. These kind of studies are powerful because each patient serves as its own control, but you gotta be really careful about the washout period to avoid carryover effects. Basically, the residual effect that can be left after the first intervention. So that's my rapid 5. I hope you enjoy it.